complex proteins and organelles involved in cellular energy production and conversion. All known cell types have evolved their own ways of generating and utilizing self-contained proton gradients. Early carbon-based life forms were presumably dependent on the naturally occurring proton gradients of the UAHM. Cells evolved internal proton gradients, which freed them from the UAHM and allowed them to seek other sources of energy and colonize the far corners of the land and seas. A hypothesis advanced by Lane and Martin suggests that escape from the UAHM required the evolution of a membrane-bound protein that can pump two types of ions, including protons, in opposite directions, called an antiporter. Several complex innovations make it possible for cells to capture energy from the environment and convert it into useful forms such as ATP. In every case, these complex structures depend on protein innovations. In addition, membrane-bound enzymes that perform sequential steps must be properly inserted into the membranes. Today, cell biologists understand a great deal about the physical and chemical properties of proteins and lipids that cause them to routinely self-organize and self-assemble within living cells. For example, membrane-bound proteins have evolved amino acid sequences called signal peptides that lead to their transport and proper insertion. When combined with natural selection, this sort of detailed biological understanding adds depth and plausibility to the UES. The endosymbiotic origins of chloroplasts and mitochondria transcend classical natural selection theory because the way that they arise cannot be described as gradual accumulation of variation. Nevertheless, natural selection clearly did transform both host and endosymbiont in each case, and the organelles have been modified and refined in an incremental fashion. The ATP synthase enzymes that produce most of the ATP in chloroplasts and mitochondria is one of the most amazing examples of micromachinery in living cells. They convert the potential energy of proton gradients into ATP by means of a channel and a literal rotor that turns, effectively transforming the potential energy of the proton flow into mechanical energy and then into the potential energy of the high-energy bond of ATP.